Hi, Martinos community. Thank you for coming. And today we're going to have Elie MacAdams. She is a researcher at the Neurotherapeutics Division. She works with consciousness, patients with deep brain stimulation, TMS, EEG, MEG. So it's going to be very interesting. Please welcome Elie.
Hi, my name's Elie and I study consciousness. So how do we currently understand consciousness? Do we think of it as a philosophy or a science? Can some people be more conscious than others? And is consciousness the same thing as awareness? And if so, are we conscious when we sleep? Are people in comas considered to be conscious? And are people under anesthesia also considered to be conscious? So, it's sort of a mystery to all of us at this point. So in my lab, we study a few different cases of consciousness. We study healthy controls while they're awake. We study healthy controls while they're asleep. And in later phases of our studies, we hope to study patients with OCD or major depression who have deep brain stimulators implanted, also while they're awake or asleep. So how do we use TMS and EEG to study consciousness? We use them to see the spread and differentiation of neural activity in response to the provoked activity from TMS. TMS creates a magnetic field to stimulate the neurons so we can observe brain functioning. EEG is a high temporal resolution of neurophysiological activity to observe the location and amount of activity. So in combination, it's really easy for us to see how where the activity comes from and how it spreads and how the different activity spreads over time. So we get real-time feedback during our sessions to find the spot in the brain with the highest TMS EEG evoked potential. So this is Patricia and I during our clinical sessions, searching for the place with the highest EEG potential. So these are all the different channels of the EEG, and as you can see, the signal averages over time, and we can see where the highest TMS evoked potential is coming from. So we keep searching until we find the best place. So in short, we perturb the system, and the circle you see up there is a representation of the TMS coil. The visualization and analysis is done with MMVT, thanks to GNOME. And then we observe. So on the right, you'll see the average butterfly plot of all the EEG channels in response to the TMS evoked activity. So also you can see the areas that were activated in response on the actual brain surface. So how do we measure consciousness once we go through these sessions? We use the PCI theory of consciousness, which is based off of the integrated information theory. What's really cool about this theory is that consciousness is considered a continuum. It's not conscious or not, it's levels of consciousness. So people who are asleep aren't unconscious, and people who are awake aren't fully conscious, but there's levels in between. So you record the brain's early reaction within the first 300 milliseconds to TMS-induced cortical perturbation with high-density passive EEG systems. You perform source modeling and pre-processing to get a binary matrix of significant sources. We only use ones and zeros for activation or no activation because we don't really care about the amplitude or magnitude of activation since we were just looking at the complexity of patterns. We then compress the matrix to calculate its information content with algorithmic complexity measures with the Lempel-Zip complexity index, which is also used for zip files to compress large patterns into very simple, easy to read patterns. And then we normalize the algorithmic complexity by the source entropy of the matrix. So conceptually, we're first perturbing the cortex with TMS to engage distributed interactions in the brain. This is the integration part of the integrated information theory. And then we're compressing that integration into the spatial temporal pattern of these electrocortical responses to measure the algorithmic complexity, and that's the information. So theoretically, the PCI should be high when we're present and awake, hopefully like right now, and low while we're sleeping under anesthesia or in a coma. Normalizing through source entropy is just a way to bound the PCI value between 0 and 1, and it's based on the binary entropy function in the information theory and statistics. Entropy is the degree of disorder or randomness within a system, so you want to divide by the maximum amount of randomness that you could possibly get to get the normalized amount. And down there is the actual calculation if anyone was interested. So to understand the actual PCI value that we get, again, it should be low for reduced interaction or loss of integration because of the matrix of activation engaged by TMS will be spatially restricted. So you won't get a lot of spread of that activity. It should be low if many interacting areas react to the perturbation in the same way because you're getting a loss of the differentiation of complex activity. So the resulting matrix will be large, but redundant and can be easily compressed into a simple pattern. This would be the kind of activation you'd see when people are asleep. But again, it should only be high when the initial perturbation is transmitted to a large set of integrated areas that react differently to this initial perturbation, which gives rise to a spatial temporal pattern of activation that can't be easily compressed or reduced into the Lempel-Zip algorithm. So graphically, the way that we represent this, ones are represented by the dark lines and zeros are represented by the white. So you average it in each time bin. So for 0.1 and 0.8, you average it. In between all of those, you're averaging it to get the initial PCI values, which you then connect the line between to see how it evolves over time. And then this is how ultimately it all looks once Patricia and I are done with the clinical sessions and I'm done interpreting the data. First, in part A, you see that we get the TMS evoked potential during the session, and then we do ICA and pre-processing, and then we get the source activation and we convert that into binary activation, ones or zeros, to then get the matrix you see in F. And then we put that into the levels of compression to then get the value that we normalize to get the PCI. And lastly, if you want to take part in this, please email me. We need more participants. So thank you.